Hello, I'm Dr. Tatjana Thomas, and today I'm going to be talking to you about the central dogma of molecular biology. So today we're going to talk about something called the central dogma of molecular biology, and this is something that's really important to all living organisms. The central dogma states that information in living organisms start out or is encoded in molecules of DNA. The information then goes into molecules of RNA. And then that information gets used to make proteins. What's interesting about this is that in all living organisms it happens this way. So in all of your human cells, information flows from DNA to RNA to protein. And in your dogs, in slime mold, bacteria, the palm trees outside, in every living organism, information is stored in DNA, but then it can get transformed into RNA and then into a protein. There are several other names for this central dogma. You can also call it information flow. Because of information flows in this one direction. You can also call it gene expression. If you want to express a gene, DNA makes up our genes. If you want to express that, it's through the central dogma. And another term you can use for this is protein synthesis. You're making proteins. You're making proteins in this process. And let's start with that. Let's look at why proteins are so important, why they're at the end of the line here. Why do you want to use information and make proteins? Proteins are really important molecules in biological systems because they're very versatile and a lot of them are structural. This is a protein like keratin. It makes up the structure of the organism. But proteins also, what I like to call do stuff. Proteins are functional. Proteins are enzymes, and they make chemical reactions in our body happen. So proteins are really, really important. And beyond that, proteins are molecules that are super, super variable. You can have an infinite number of different proteins. And you are who you are because of the exact number and structure of your proteins. It's not your lipids that differentiate you as a species or as an individual, not your sugars, but it's your proteins that make you you. All right, so let's look at this central dogma, this information flow, a little bit closer, and we'll start by looking at this first process, getting information from DNA into RNA. And that process is called transcription. Transcription. And in transcription, you're going from the DNA to the RNA language, and both of these languages have 4 variables in them, 4, if you will, letters, and those letters are nucleotides. The nucleotides, these letters in DNA and RNA, are very similar to each other, and like I said, there's 4 of them. So you're going in a language with 4 variables into another language of 4. This is your A, C, T, and G in DNA, and very similar in RNA. So kind of to see why this is a fairly simple process, I'm going to erase this. Let's look at an analogy to the English language. A phrase like good fortune. The arrangement of variables of letters in the English language, this has meaning to us. Going from DNA to RNA is like taking something from print to cursive. So if you take this phrase, which has meaning, and convert it into cursive, that's similar to going from DNA to RNA. Now you might ask yourself, why put the information in RNA in the first place? If you want to make protein, if proteins are so important, why not take the information from DNA and put it directly into a protein? Why go through this, what you will middleman step? Why make this middleman molecule, this RNA molecule? And the reason for that is that the DNA is very precious, and you do not want to damage your DNA. And it is similar to if you're studying an ancient book that was maybe written in the Middle Ages, and you check that out. Let's say your local library has this book. If you want to study that book, you're not going to go and check it out and take it home and sit at your kitchen table and drink your coffee and study this book. It is way too precious. You only have this one copy of it. It is way too precious to do that. Instead, what you would do, you would make copies of the pages at the library, keep the book nicely safe, protected at the library, and then you take the copies home, and then you study and do something with the copies. And that's what we're going to do here. The RNA are the copies of the information in the DNA. All right, so let's look at the second step. The second step from taking information from RNA to protein is called translation. And sometimes students will mix up those two words because they sound very similar. But when you look at transcription, there's a C here. Translation, there's an L. So C always comes before L, so transcription always before translation. So just remember that. Now, proteins are not made up of nucleotides. They're made up of amino acids. So amino acids are the variables that are in proteins. And there aren't just 4 amino acids, but there are 20. So now you're going from this language that only has 4 variables to a language that has 20 variables. So that is very different, and because of that, this is a much more difficult process. It's a process that occurs in ribosomes. And it's almost going from one language to another language. So it's like going from English to Chinese. If you want to get this information across in Chinese, it will look very different. And I don't have that memorized, but it would look like this. Somewhat like this. This is my best interpretation of it. But when you're going from RNA to protein, it's like translating, like going from English to Chinese. You have that same information that's being translated, but it looks very different. And because of that, that process is a lot more involved. All right, so in summary, the central dogma shows us that information in living organism flows from DNA to RNA to protein, and that the first part of that process is called transcription, making the RNA, and the final part of that process is called translation, making the protein. But why is this important? Why is this the central dogma? It's important because in all biological systems, it occurs this way. You have information stored in DNA. That's how you store information in living organisms. In the digital world, we store information in 0 and 1s. That's storing information there. Out of the 0 and 1s, you make your computer game, your virtual digital world. Out of the 4 variables of DNAs, your ACTs and Gs, you make life. And this is, again, how all organisms do it, and it's been done like this. Never been done any different. All right, well, thank you for watching this little explanation on the central dogma, and have a nice rest of the day. Thank you from Dr. Thomas at Paradise Valley Community College.